Thank you for the opportunity to present. My name is Doug Storm, and I will be presenting on how the mode of birth delivery and pediatric age affects the pediatric urinary microbiome. We have no disclosures. We have previously described the existence of the pediatric urinary microbiome in both males and females. Prior studies have suggested that age may affect the gut microbiome in children, and also mode of birth delivery by either cesarean section or vaginal delivery may also affect the pediatric gut microbiome in different ways. To date, though, these effects have not been studied in the pediatric urinary microbiome. Our study aims were to evaluate the effects of a child's age on the pediatric urinary microbiome, and also to compare the pediatric urinary microbiome by mode of birth delivery, either through cesarean section or by vaginal delivery. To perform the study, we collected samples from children anywhere from 0 to 18 years of age. The samples included a catheterized urine, as well as swabs from the perineum, the urethra, the vagina, and the foreskin. These samples were all collected on children undergoing procedures which required a general anesthetic. They needed to be free from antibiotics for at least 3 months and could not have a current urinary tract infection. The samples were collected from children either from the University of Iowa or the University of California, San Francisco, and they were shipped overnight to Loyola to be processed. Here they were set up for ECOG or for 16S sequencing. To date, we have collected samples from 86 children over a wide age range. You can see that about 50% of them are male and another 50% are female. And in terms of the percent with a positive urine sample, it was around 60%, which is very much in keeping with the with studies in the adult population. In terms of the type of bacteria that we identified, you can see that there's a wide variety of bacteria which we identified in these children's urine. Most of them are very non-traditional bacteria, meaning that these are not common bacteria causing urinary tract infections. You can see that the most common bacteria identified were staph and strep, as well as enterococcus. In terms of the effect that a child's age has on the microbiome, you can see that the majority of the bacteria actually appear shortly after birth. Some of these bacteria will disappear around the age of toilet training, while the majority will persist throughout childhood and into their teenage years. You can see that there are some bacteria which occur as the child begins to go through the age where we would expect them to toilet train. Some of these bacteria then will disappear once they go through their teenage years, whereas other bacteria will persist or actually arise mostly in the teenage years. In our study, 70% of patients were born via vaginal delivery, while only 30% were born via cesarean section. And you can see that there's an equal breakdown between males and females with the two groups. Taking a closer look at how the mode of birth delivery affects the pediatric urinary microbiome, you can see that there are some bacteria which are shared between those born via cesarean section and those born vaginally. There are only a few bacteria which are isolated to those born via cesarean section, where there is far greater bacteria which are isolated to those born via vaginal delivery, and this is much more diverse as compared to those born via cesarean section. So taking a look at this as a whole, you can see that there's a much more diverse microbiome in children born via vaginal delivery versus those born via cesarean section. In conclusion, the pediatric urinary microbiome appears to develop early in life and has some changes which occur throughout childhood and into the teenage years. There appears to be a difference in the bacteria constituting the urinary microbiome in those born via cesarean section and those born via vaginal delivery. And those born vaginally appear to have a much more diverse bacterial makeup of the urinary microbiome as compared to those born via cesarean section. Certainly, additional sample collection is ongoing to help further delineate these findings.